We had talked about the immersion effect. In that effect, we understood that it is not just one pigment that is playing its role. There is a possibility that different pigments would be involved. Now we are going to jump to the next topic, that is about the spectrums. Two spectrums we are going to study. One is the absorption spectrum, and corresponding to that absorption we have the action spectrum. Now, in case you are hearing this thing for the first time and it goes over your head that what this must be, let us see what the term itself has in its words, the meanings that we can decipher from it. First is spectrum. Now, spectrum, whenever this word comes, do keep in mind that you are dealing with white light which can be broken down into different wavelengths, and those different wavelengths correspond to different colors. One thing. Now we would be dealing with those different wavelengths of white light. That means we are having a spectrum. Now the other word in both the cases, one is absorption and the other one is action spectrum. Now you're clear with spectrum? Let's see what absorption spectrum has to say. As the name suggests, there would be absorption of something. Now what that absorption would be, because it is a spectrum, because it is dealing with white light, because it is having different wavelengths in it, the white light I am talking about, the absorption of different wavelengths of the white light by a surface when it is depicted by a curve, alright, that would be known as action spectrum. Not action spectrum, absorption spectrum. I repeat it again. The absorption of the different wavelengths of the white light as represented by the spectrum, if they are taken in account in the form of a curve, that would form the absorption spectrum. Okay, and when we talk about absorption spectrum, we are saying here that it is the absorption by a particular substance. Absorption of what? Of different wavelengths of the white light. So here we have the representation of almost a region known as Vibgyor, where we have the wavelengths ranging from violet to red. This is the wavelength in nanometers. And these different colors are depicting different pigments which are present inside the leaves. Okay, this entire absorption spectrum is obtained by an instrument known as spectrophotometer. If you can remember it, it's good that this is the instrument that we use for getting the absorption spectrum. Now this absorption spectrum shows us that how each photosynthetic pigment that we are considering is going to absorb the wavelength, which wavelength it is going to absorb and where the absorption is maximum. What you see over here is that I have used three colors to represent. This blue part represents chlorophyll A. Chlorophyll A is a photosynthetic pigment. Then the green part represents chlorophyll B, and the red part represents carotenoids, all the pigments except the chlorophylls. They would comprise the carotenoids, specifically beta carotene. Now this is what is the absorption spectrum. This first one is the absorption spectrum. As you can see, we are showing the wavelengths over here, and this region as it is higher, it shows the absorption of light, that means absorption of energy by the particular pigment at that particular wavelength. So let us consider the chlorophyll A pigment that is represented in blue color. It absorbs maximum in this region. Okay, this is the maximum absorption that it is showing, that is the violet and blue region, that violet and indigo region. As we move further, the wavelengths are increasing. The absorption over here, it declines to a great level, and it later on rises in the red region again. So this chlorophyll A shows the absorption in the violet and the red regions where it is absorbing the maximum wavelengths for taking up the energy. So this shows the absorption spectrum of the chlorophyll A pigment. Similarly, in the case of chlorophyll B, what you see, the absorption is maximum in this blue region, then it comes sharply declined, it goes, and then it is like in a wavy pattern, very low absorption takes place, then it rises again in the red region, similar to what chlorophyll A is doing. So chlorophyll B has this absorption spectrum. Talking about the carotenoids, which are represented by the color, they are reddish orange color, and their region of absorption ranges between the violet and blue. Then the absorption stops. They do not absorb in the red region. That is why they reflect it and they are reddish in color. So this is the absorption spectrum of 3 different pigments that we have talked about. Now, what is the relevance of this absorption spectrum that we get? We get an action spectrum as well. When we have a good absorption spectrum obtained by our experimental evidences, we according to that observe that corresponding to these wavelengths where the absorption is maximum, the rate of photosynthesis is also high. So the action spectrum, contrary to the absorption spectrum, it shows the rate of photosynthesis depicted in a relative manner. So while the absorption spectrum was representation of the wavelengths absorbed by different substances, okay, and we take those substances to be pigments, the action spectrum is the activity. This is the absorption that the wavelength has absorbed, the wavelength is taken up, it is being absorbed at different wavelengths by different pigments, that was represented graphically. Now we have to represent the action, that action over here, because we are dealing with the topic of photosynthesis, the action over here would be photosynthesis. Now how that photosynthesis is observed, it is observed as rate of photosynthesis, okay. Now this rate of photosynthesis, when it is depicted relatively according to different wavelengths. At what wavelength do we have which rate? That is what is action spectrum. Like if you have the wavelength that ranges between violet and indigo, what is the rate of photosynthesis at that point? Then as you move through the spectrum, what would be the rate of photosynthesis? That would be represented in the action spectrum that we have over here. Now, unlike the absorption spectrum, which shows dark, steep uprises and then sharp declines, this photosynthetic rate is not having that much declining, these dips as we have in the action spectrum. Rather, it is having almost a little bit elevated rate. Then we have almost constant rate where there is minimum absorption. Even at that point, you can see those wavelengths only are represented over here. We do not have declined photosynthesis as it should be expected from here. Get my point? The rate of photosynthesis is almost equivalent. It sharply declines after red. Okay, the red wavelength, that is known as red drop as we call it. Now, what is the reason that this region of, I would repeat, what is the reason that this region of the graph does not decline corresponding to this? We talked about that it is ultimately the photosynthetic pigments that take up the energy and they are responsible for the photosynthesis. Over here we see that there is least energy absorbed over in this zone, but the photosynthesis is not declining like the way it has shown over here. The spectrum should have been lower. The reason behind it is that all these pigments are working together, and that point you will understand when you study photosynthetic unit, because all these pigments, they would not be working individually, they would rather be working as units. And this is the reason, this zone, why it shows photosynthesis, though the absorption is minimum over here. This is the reason that why people, or you can say great minds, they got into studying that what is actually happening. It would not be a single pigment that would be working, there would be a group of pigments which would be involved, and then they came to the conclusion that there are photosynthetic units which are involved, which we will study later on. But for now, what you have to keep in mind that there is an absorption spectrum, and contrary to that we have an action spectrum which shows the relative rate of photosynthesis, keeping in mind the absorption of the different wavelengths. The rate of photosynthesis is a little bit slightly different because we do not get the same dip as in this case. Now, who proposed this basically? Who came with the idea of this action spectrum and how it was experimentally proven? There was a scientist known as T.W. Engelmann. You have to remember his name, and Okay, he was the scientist. He is responsible for telling us experimentally that it is the action spectrum actually is there. You see that rate of photosynthesis corresponds to this action spectrum. What he did was he used a green algae, and same way as we have Vibgyor, the similar manner as the action spectrum is shown, he used the green algae and he illuminated the green algae in the same way using a spectrum. Okay, some part of it was having violet wavelength, indigo, blue, same as we have the white light spectrum. Now what happened after some time, he also used certain bacteria which feed on oxygen. So he took a culture in which he took a green algae, and inside it he put some aerobic bacteria, and then he illuminated that culture with this white light that was scattered white light. It was in the form of the spectrum. Now what was observed after some time, that the bacteria, they got accumulated at these regions. Now you know the bacteria is feeding on oxygen, they are aerobic bacteria, so the more concentration of bacteria was found in these regions and least was here. So he in a way proposed the action spectrum on the basis of evolution of oxygen. Okay, so you have to remember these things, that in 1882 there came somebody by name of T.W. Engelmann. He came with the idea and showed us how action spectrum is there, not action spectrum, absorption spectrum actually is there by identifying the rate of oxygen evolution using anaerobic bacteria. He proposed that in this zone and in this zone the rate of photosynthesis is highest. Okay, these regions, they do not produce much oxygen because it depends on the rate of photosynthesis. How much photosynthesis is taking place, that much would be evolution of oxygen, and that was proved by using the bacteria. And from the absorption spectrum we get the action spectrum as well. These are the two terms that you have to remember before you set up yourself into studying the actual mechanism of photosynthesis, how it takes place, what are the units involved in it.